Hi everyone, my name is Karen Corbett. I'm a postdoc at Monash University, and I'm going to be telling you a little bit today about my work with cyclic peptides. So cyclic peptides are a fast-growing market in the pharmaceutical industry, and many of these have pharmaceutical activity by binding to some target protein of interest. So both the binding to this target protein, as well as the permeability of the cyclic peptide through the cell membrane, can be really influenced by the conformational ensemble that the peptide can take. That is, what 3D structures can the peptide form, and what are the relative populations of those 3D structures? So as an example, I have a cyclosporin A, the cyclic peptide drug, to the right. So cyclosporin A in water has several different conformations, some of which are non-active. As an example, I have this one here, and one protein binding active conformation that is able to bind to the protein target of interest and have pharmaceutical activity. So as you might imagine, the amount of the active conformation that you have is going to really influence your pharmaceutical activity. So for instance, there's some cyclosporin A analogs that have increased protein binding conformation population, and then thus increased pharmaceutical activity. So if we really want to understand how our cyclic peptide drugs are working, we need to understand, one, what conformations do they take, and two, what are their relative populations. So the question I'm going to pose for this talk is, how can we study the conformational ensembles of cyclic peptides? Historically, this has been pretty difficult to study experimentally. And one of the major issues is that a lot of the interesting conformations are low population, and thus have very low signal to noise if you're trying to study them experimentally. So going back to the cyclosporin A example, our active protein binding conformation has really high free energy or low population relative to the other 9 non-binding conformations. And in fact, due to this low population, the protein binding conformation of cyclosporin A in water has still not been solved due to this low signal to noise issue. So another way to try to attack this is with computer simulations. So in particular, I'll be talking about molecular dynamics, or MD simulations. So MD simulations are great in that you can have every single atom in your system of interest represented, and then you can propagate them with Newtonian dynamics. So this includes both your cyclic peptide and the surrounding solvent. The issue with MD simulations is that you have to use a time step around a femtosecond. So a lot of the conformational changes that we're going to be interested in are much longer timescales. So for example, peptide bond cis-trans isomerization, which occurs for a lot of these cyclic peptides, is around the minute timescale. So to get one isomerization event, we would have to do over 10 to the 15 computations, which is just not feasible with current computing power. So another way to look at this issue is in terms of free energy. So you can see in the diagram here that MD simulations can get stuck in local free energy minima for long periods of time, because it just takes too long to overcome the free energy barriers to look at other local free energy minima of interest. So we can see what this looks like with this movie. So you can see the peptides moving around, but the peptide backbone conformation, which I'm circling here, is not really changing that much. So this peptide is stuck in this local free energy minima and not able to explore other interesting conformations. So one way we can get around this is a special kind of MD simulation called temperature replica exchange MD simulation. And I'll be calling this T-Rex simulation for the rest of the talk. So for T-Rex simulations, you simulate a whole series of MD simulations in parallel at different temperatures. And then periodically you attempt to exchange between two replicas. So whether the exchange is accepted or not, it depends on the metropolis criterion. And that is, how close are the potential energies and the temperatures between these two replicas? Then, if the exchange is accepted, then you swap coordinates and rescale momenta for these two replica pairs. Then, after the exchange attempt, you carry on with your regular MD simulation until the next exchange attempt. So the reason why we're doing this is if you're stuck in your local free energy minima at your lower temperature replica, you can exchange with a higher temperature replica, which has a flatter free energy surface, and then you can travel to new interesting local free energy minima in the higher temperature space and exchange back to your lower temperature replica. So in this way, you can overcome these free energy barriers by exchanging to a higher temperature, moving to a new interesting region, and exchanging back. So we can clearly see the difference in what this looks like for our simulations with this movie here. So this is a T-Rex simulation that's the exact same length as the MD simulation in the previous slide, but you can see clearly this peptide is able to sample a whole other range of conformations that the MD simulation could not. So the peptides that we're going to be looking at in particular are two low-key peptides, So Loki-3 and Loki-2. So these are cyclic hexapeptides, and the only difference between these two are highlighted in red, and their only differences are in chirality, where D amino acids are indicated by the lowercase letters, and L amino acids are indicated by the uppercase letters. So we're going to be looking at the conformational ensembles of these two peptides in water and chloroform using T-Rex simulations. So I'll be starting with the Loki-3 peptide in chloroform. And I want to just note that what we're going to be trying to do here is we're going to be starting with a really bad starting structure to make sure, can we still predict the experimental structure? So what I have shown here is a reduced representation of our simulation starting structure, just the peptide backbone for making it easier to look at the differences. And our simulation starting structure is in cyan, red, and blue, and is overlaid on top of the experimental NMR structure. So you can see these don't match very well at all. And in fact, our simulation starting structure has an incorrect cis-peptide bond conformation. We know from experiment that this particular peptide should have all trans peptide bonds. So with this bad starting structure, we can follow the progression of the cis-peptide bond with its corresponding omega dihedral angle with simulation time. So you can see at the beginning of our simulation, we're all in the cis-peptide bond state. And then the population of the cis-peptide bond starts to decrease with time, and the trans-peptide bond population increases with time, until by the time our simulation is converged in this last region here, we're in the all trans-peptide bond conformation in agreement with experiment. So not only that, but we have one major conformer in agreement with experiment, and 99% of our structures from our simulation are within 1 angstrom Armistead NMR structure backbone. So to reiterate, we start with a really bad starting structure. With 0 input from experiment, we're able to predict the experimental NMR backbone structure. I also want to reiterate that this would not be possible with regular MD simulations, as mentioned at the beginning of the talk. Cis-trans peptide bond isomerization takes place in the minute time scale, which is just not feasible for regular MD simulations. So next, we want to look at the Loki-2 peptide in water. So again, what I have here in cyan, red, and blue is our T-Rex simulation of Loki-2 in water, and it's overlaid on top of the experimental NMR structure for Loki-2 in water. So you can see for this one, again, it for the most part matches the experimental NMR backbone structure very well. We have about 87% of our structures within 1 angstrom Armistead, the NMR structure backbone. However, you might be able to see in the movie, there's some structures that pop up once in a while that don't match the NMR structure backbone. So we wanted to look at this in a little more detail. So what I have on the plot to the right here is our normalized count with respect to the Armistead to the NMR structure backbone. So you can see most of the structures are within this 1 angstrom Armistead cutoff that match very well to the NMR structure backbone, where I have a representative snapshot here. However, some of these structures in this region here don't match very well to the NMR structure backbone, where I have a representative snapshot here. So we wanted to look at this in a little more detail. So we found actually that our NMR spectroscopy results show that there is a low population conformation in addition to this major conformation here that has a cis-peptide bond, where this major conformation has an all-trans peptide bond. So NMR spectroscopy was not able to resolve this low population conformation due to this aforementioned low signal noise issue, but they were able to determine that it has a cis-peptide bond. So really interestingly, this conformation here has a cis-peptide bond in that aforementioned region. So it's really cool that our simulations are matching up with these, the minor population information that we have from NMR spectroscopy. And we're also able to have an atomistic structure as an estimate of this minor population conformation. So to summarize, our T-Rex simulations were able to predict the NMR structures representative of the most populous conformations. Our T-Rex simulations were also able to determine low population conformations, such as this one, that match NMR spectroscopy cis-trans estimates. So in answer to the question I posed at the beginning of the talk, we think that T-Rex simulations are a great way to look at the conformational ensembles of cyclic peptides. And with that, I'd like to thank Sue Kwan for her NMR spectroscopy work, Lonza Pharma and Biotech for funding, and as well as American Australian Association, Massive HPC Facility for computing time, and David, Colin, Lee, and Huel for supervising my work. And with that, I'd like to thank you for listening. And also, I'm going on the job market, looking for a postdoc. If anyone is hiring, please send me an email.